Okay, so this is chapter 19. We're going to be taking a look at here. It's going to be cell division. And so I will move somewhat briskly through this particular chapter. So, excuse me, sorry. So first of all, we're going to take a look at what we're actually separating in terms of cell division, so our chromosomes. And so the chromosomes are going to be found in the nucleus. This is where our DNA is going to be found, and our DNA is going to be bundled up and structured into structures called chromosomes. And this is going to give us the instructions to pretty much do anything and everything that's necessary in your body. And so within this are going to be genes, and they're going to be made out of DNA. Now, chromosomes also don't just have DNA in them, they also have proteins in them, and this will help with organizational structure. And it forms a structure called chromatin, where you have these like little protein complexes that look like little drums, and then you have DNA basically wound around them. So that's basically what chromatin looks like. Now, in humans, we have 46 chromosomes in each of our cells, 23 pairs, so we have 2 of each. The first 22, 1 through 22, are what's referred to as autosomes, and that's in both males and females. And then the other 2 are going to be what's called the sex chromosomes, so X and Y. And so when you take a look at the sex chromosomes, X and Y, ultimately, males will have an X and a Y, and then females will have 2 X's. And the reason for that is that on the Y chromosome is a specific region called the sex-determining region of the Y, SRY. And this is essentially what causes testes to develop from embryonic tissue. In the absence of the SRY gene, those embryonic tissues develop into the female sex organs or reproductive organs. And so the Y confirms maleness in human beings. Now, in order to see the organization of the chromosomes, we have a particular type of a test called a karyotype, which is simply a display of your chromosomes, everything that's basically involved in a cell. So when a cell divides, that chromatin sort of squeezes together, condenses to form tighter and tighter chromosomes. And then these chromosomes can be harvested, and then they can be stained. And so they'll create these light and dark bands that are specific to the individual chromosome. And then we can sort these all out by the chromosome number, and it turns out to look something like this. So basically, you can see after staining, they have a specific staining pattern. So each of the chromosomes consists of what's called sister chromatids. So these are the individual little copies of a gene connected at a central location, which is called the centromere. And you have one from mom and you have one from dad. These are what's referred to as homologs. That's the reason why you have 2 of everything. And so in this particular situation, you can see how you essentially have collected the 2 ones, mom 1 and dad 1, and you collected the 2 2s, mom 2 and dad 2, mom 3, dad 3, mom 4, dad 4, and so forth, all the way up to 22. So you notice you have at this particular point now 44 altogether. And then you're going to get an X and a Y. And so in this particular case, the Y chromosome is coming from dad, the X is coming from mom. And so this gives you number 45 and number 46. And that's how each of our cells has 46 chromosomes in it. We basically have 2 of every pair, 1 set coming from mom and 1 set coming from dad. So then, ultimately, when you take a look at segregation, you're going to be looking at the process called mitosis. This is basically cell division, and this happens as soon as the egg is fertilized. It becomes what's called the zygote, which is a diploid cell. And the zygote will have mom and dad's chromosomes in it, and they will start going through a series of mitotic events, creating 2 genetically identical daughter cells. So that basically means that every single cell in your body is what's called diploid. That means it has 46 chromosomes, or basically 2 sets of each chromosome. So in a dividing cell, each of your chromosomes has 2 copies called sister chromatids. So for instance, normally, before you go through replication, you have a chromosome, there's your centromere, and then what happens is when you go through replication, for the purpose of division, you're going to basically create a copy of that chromosome, but it's still going to be tethered to the same centromere. So these guys now are what's referred to as sister chromatids. Just to avoid confusion with the term that's already been used, which is chromosome. And that's happening on both the mom chromosome and also the dad chromosome. Genes then are basically little hereditary packets, if you will, and their job is to control a particular trait. For instance, there's a gene for eye color, there is a gene for hair color, and so forth. And so altogether, all of these genes essentially creates all the instructions necessary to essentially create or build your body. Now, centromere, when you take a look at a chromosome, is essentially a constriction point in all chromosomes that holds your chromatids together. And so during cell division, what happens is you separate your sister chromatids apart, forming new independent chromosomes in the daughter cell. So for instance, if I have, let's say this is one sister chromatid attached to the other, what's going to happen is I'm going to pull one set of sisters to each direction. And so this set of sisters will basically move to one side, and this set of sisters will move to the other side. And they'll become the new chromosomes of that cell. So here you have basically two identical cells. And now, once they're in there, they can function as fully functional chromosomes. Okay, so let's go ahead and take a look at cell cycle. So in cell cycle, basically you have two main parts, interphase and cell division. And that's the reason why they call it interphase, because it's an intervening area between cell divisions. So before cell division occurs, typically what's going to happen is in a non-dividing cell, chromatin is kind of spread all over the place. And it's kind of a tangled mass of threads and just basically kind of like you're using it, right? So it's basically actively being used DNA. And then what happens then is you have to sort of package that down, and then you have to get that ready to move. Now, once you are in the cell cycle, and the cell cycle, you're always in it at some particular point. There's no such thing as a cell that's not in cell cycle. It's just in various parts of cell cycle. Most of the time that the cell is existing, it's mostly an interphase, this sort of intervening period between cell divisions. And during this particular phase, it's just the cell living its life. It's basically just doing its usual functions, kind of doing what it does. The organelles are doing what they do. And then ultimately, to get ready to divide, it has to grow. That is, it has to get larger. It has to double everything, right? So remember, the mantra of division is basically duplicate and then separate. So it has to duplicate everything, not just DNA, but also duplicate the organelles. And it has to also duplicate the DNA. And that's the prepare for separation or for division. Now, generally speaking, this is all events of once that are happening in different places in interphase. For instance, interphase has three main subphases to it, G1, S, and G2. And you can actually draw the entire cell cycle in a big kind of a wheel chart. So beginning first of all with G1, this is sort of like the cell's growth period. This is like a cellular childhood as it's getting larger. If a cell decides to just not do cell division, then what it can do is it can basically spin off into sort of like this little side state called G0. This is just a functional state where the cell just indefinitely does whatever its job is. Now, if it decides to divide, then it's going to go into the duplicate and separate mantra. So the first thing it does is duplicate its DNA. That's the S phase. And then it'll go into the G2 phase where basically it will duplicate its organelles. So you're duplicating everything here. And it prepares for division. Once you get into the M phase, this is mitosis, then you're basically separating the nucleus. This is where you get those two sisters pulling apart from each other. And then cytokinesis is the separation of the cell and the organelles. And then once you do that, you have a newborn baby cell, which then itself enters G1 and is ready to grow up. And so on and on it goes as a cycle. So in G1, basically, this is kind of normal functions. It's your cellular childhood. You're just kind of growing up, doing your thing. And so ultimately what's going to happen here is you're going to be, just kind of doing your general business. And if you commit to the cell cycle, you're going to be getting yourself ready for DNA synthesis. That's the next stage. S phase is basically DNA replication. This is where you double your chromosomes. You make an identical copy of your chromosomes. This is where you create the sister chromatids that are attached to the same centromere to each other. And then after S phase, you go into the next phase, which is G stage, or the G2 stage, or the G2 area. And so this is where you're getting ready for cell division. So you're making all the proteins that you're going to need for that. Also in this area, you're doing a lot of DNA repair and different things. You're getting ready for that division. And so when you take a look at a cell's life, it's spending a lot of time in interphase, and how much time it spends there can vary wildly, actually, between one cell and another. So for instance, nerve cells, muscle cells, these typically aren't going through cell cycles, so they don't divide. What does that mean? So that means if you actually damage a muscle cell, your muscle cell's lost. If you lose a nerve, you damage a nerve, then you don't get that nerve back. So these guys are kind of in permanent arrest. They're in this constant sort of G0 state, this sort of functional state. And so they kind of just are terminated there. And so they don't divide, which is important because that basically means these types of cells you have to be very careful with, because if you damage them, then you're just going to be living with the damage and you're not going to be able to get those back. Embryonic cells, however, which are actively dividing and dividing very, very quickly because they're trying to build a little embryonic body, don't spend a whole lot of time in G1. I mean, they just go from one division to the next, to the next, to the next, because they're just trying to make as many cells as possible so that you can build your big complex body. So after interphase, then is going to be M phase for mitosis and then cytokinesis. So mitosis is specifically looking at nuclear division. And so, so ultimately at this particular point, you're going to be creating two different nuclei. You're going to be separating one nuclei with twice as much material in it into two nuclei with the normal set of material in it. And then after that, you're going to divide the cytoplasm and the rest of the cell, which is cytokinesis. Now, mitosis and cytokinesis, basically, these guys are kind of overlapping. And so this can oftentimes continue and kind of continue to go on. And so on one end of the spectrum, you've got mitosis, which is creating new cells. On the other end of the spectrum, you have apoptosis, which is programmed cell death. So if you don't need any cells anymore, then basically you go through apoptosis. Or if you have a lot of damage in a particular cell, then you can also send it through apoptosis. A good example of that is embryonically is a lot of times, if you take a look at your fingers, when you're an embryo, when you're just a fetus, you'll have like a little bit of webbing in between your fingers, but that webbing at some point will go through apoptosis, programmed cell death. And so those cells will die out. So when we're born, we don't have webbed fingers because those cells die out. So then the cell cycle is also very controlled and regulated. It's controlled by what's referred to as checkpoints. So checkpoints basically are stopping areas. So they're basically pauses in the cycle. And basically what they're looking at is they're kind of just double checking for quality. So it's kind of like your quality control check, making sure everything is okay. And you have three major checkpoints. You have a G1 checkpoint to make sure that all that growth and that sort of stuff is going well. You have a G2 checkpoint where you're oftentimes looking at the integrity of DNA, if your DNA is repaired. And you have what's called a mitotic checkpoint, which is sort of stopping right in between, kind of arrested right in the middle of mitosis. And so this kind of gives you one last checkpoint just to make sure everything's okay before you separate your nuclei. Now, generally speaking, these cell cycles are going to be controlled by external factors like hormones and growth factors, which are typically stimulating mitosis. Now, if you fail in cell cycle control, or if the cell becomes out of control, that is to say they're not listening, then that can create what's referred to as unrestricted cell growth, which can ultimately lead to cancer, because cancer basically is essentially a disease of uncontrolled cell growth. So that's kind of what it looks like. So in G1, you're going to see your G1 checkpoint. This is basically making sure that everything in your cell is growing and that your cell is normal, that everything is good. As you go through S phase, you're going to stop again at G2. This is making sure that your DNA is replicated properly, that everything is good. If you need DNA repair, then it sort of stimulates that. And you're ready for mitosis. And then right in the middle of mitosis, you're going to go through what's called the spindle checkpoint, which is essentially where you're checking to make sure that all of your chromosomes are appropriately attached, so that when you go to pull them apart, they're going to pull apart in a very sort of a symmetrical way, so that you get good, even segregation. Now, this checkpoint, ultimately G1, is essentially your commitment point. This is where you're committing to divide. At this particular point, you're answering a question: do I divide or not? So if you don't pass this checkpoint, then you go into your functional state, right? That's just where you do your thing. However, if you do go through this checkpoint, then you're going to be committed to the rest of the cell cycle. So then, at this particular point, you can also check to see, even though you haven't gone through replication yet, you can check to see if there's any damage in your DNA, proteins, if there's anything abnormal in the cell, for instance. It's kind of a check for normalcy. And a very important protein in cell cycle control called P53 will do that. So it'll stop G1, and it'll basically determine the quality of the cell, if you will, to see whether, to make sure everything is okay. If there is DNA damage in this particular cell, then it will allow you to be able to move forward, to clean it up, to fix it, and then to move forward. The G2 checkpoint, this is basically where you stop the cell and it makes sure that your DNA has been replicated. It makes sure that all the chromosomes are duplicated before doing replication, or before doing division and mitosis in the M stage. And then it's also checking for DNA damage, right? So if there's DNA damage, it'll stop the cell cycle, allow time for that DNA to be repaired so that it can be fixed up. And then the mitotic checkpoint basically is a stoppage between what's called metaphase and anaphase. This is basically where you're making sure that the chromosomes are all correctly attached, that when you go to pull on them, they're going to pull in a good, symmetrical, organized way. So your external control that we see is essentially coming from outside. So these are signals coming from outside telling the cell whether or not it needs to divide or not. So typically these are hormones and growth factors, and these are typically what's stimulating division. Normally what's going to happen is these growth factors will come in, they'll bind to a receptor that's on the plasma membrane of a target cell. And this receptor will then communicate into the internal part of the cell, relay that system to the proteins inside, and that will basically tell that cell to do whatever it needs to do. This is a process called signal transduction. Now, it's the important thing to know is that for every dividing cell population in your body, they are under the control of a growth factor. So these external signals are the ones controlling when and how much to divide. So then, at the very bottom of the signal transduction pathway are the actual genes that will be responsible for moving the cell cycle forward. And typically, they have two different types of genes. One's called a proto-oncogene. This will typically be a gene that stimulates the cell cycle. And then you have what's called a tumor suppressor gene. This is a regulatory gene that will tend to inhibit the cell cycle. So one will stimulate the cell cycle, the other one will inhibit the cell cycle. And so this is kind of what it looks like. So here you can see a receptor in the target cell, and then your growth factor will come in and bind to that receptor that communicates to the cell via a signal transduction pathway to basically to get ready for cell cycle. So normally a target molecule will come into the nucleus and activate and turn on genes that will be necessary in this pathway as it comes out and translates new proteins that will basically stimulate cell cycle. So that's basically how you move cell cycle forward. Now let's go ahead and take a look, a closer look at mitosis and sort of the events of mitosis. So mitosis itself is going to basically create new cells. Okay, this is how you go from a single cell to a full body. And so this is growth. And then once you stop growing, then you're going to be replacing your cells as an adult. And so that's what most adults are doing right now. So their mitosis is a replacement strategy. Generally speaking, during mitosis, your cells will divide. The dividing cell is called the parent cell. The offspring are called the daughter cells. So for instance, the parent cell is the one I wrote before, who goes through mitosis to create two identical daughter cells. So this is the parent, and these are the daughters. So this is called duplication, mostly because you're duplicating the number of cells. So for instance, in healing, you need mitosis in order to be able to make the new cells that will essentially close up the wound. Now, when you take a look at mitosis, it all starts by duplication. Remember, the mantra of mitosis is to duplicate and then separate in that order. The first place you start is the duplication of your DNA. That's going to be happening in the S phase, as your chromosomes are essentially going to go from single chromosomes to two sister chromatids attached by the same chromosome. So during S phase, you're doing DNA replication. And you're going to basically grow a new, or you're going to make a new chromosome attached at the centromere. And so it's going to look something like this. So once you actually do replication, you're going to be adding that sister chromatid. In the reverse direction, you're going to be pulling those two sister chromatids apart during division to give you one. So that's a very nice graph to kind of see how this is back and forth. Ultimately, when you begin mitosis, it doesn't just begin with duplication, but once you actually have your duplicated chromosomes, you have to condense them down into tighter and tighter little packages. And then you go from a chromatin structure, which is kind of loose and just sort of like little fibers all over the place, to a more condensed chromosome, which is more packaged and more condensed. And so this will allow you to be able to move your chromosomes to each daughter, where each daughter gets ultimately a complete set of chromosomes. And so they are genetically identical to each other and to the parent. That's the important thing. In the case of mitosis, you're basically making more of the same. You're not looking at differentiating yet, although some of these cells will go on to become differentiated, but they'll become differentiated in a different sort of a fashion. That's beyond the scope of this chapter because that gets into embryological development. So within a chromosome, you have, within a cell that's important for mitosis, you have the development of what's called the mitotic spindle. The spindle is essentially a structure that begins with the organelle, the centrosome. Now, the centrosome is literally a microtubule organizing center of the cell. That basically means that this is an organelle where you're going to be able to extend out and build microtubules in sort of this big array and sort of be able to kind of cast out all these microtubules all over the cell. So they will duplicate, and then they'll separate into an antipolar position. So they'll be basically opposite each other. And then as they grow out their microtubules, they'll create what's called the microtubule and the mitotic spindle. And so this is basically this big network of spindle structures. So for instance, here's my other centrosome. So it's also shooting out microtubules in all directions. And ultimately what they create is this kind of large birdcage looking structure of microtubules, which is called the mitotic spindle. So this would be the mitotic spindle. Now it's this structure that's going to be allowing you to be able to move your chromosomes and separate your sister chromatids to each pole. First thing they have to do is attach to the individual centromeres. Now, oftentimes, when they're first growing, they oftentimes are referred to as asters because the little centrosomes, as they start to shoot out little short microtubules, before they've grown out to full spindle, they look like little asters. But in their fullness, you'll see them more as spindle poles. Now, each centrosome ultimately has a set of smaller structures called centrioles, which are short cylinders of microtubules. So if you take a look at a centrosome will basically have this little tube that are perpendicular to each other of microtubules. And it's from these tubes that you can stack your microtubules onto and grow those microtubule fibers out in just pretty much any direction you want. And that's kind of how you can get the development of the spindle. Now, with that in place, then we have a structure to be able to separate out our DNA. We've got a duplicated DNA. Now we can talk about the phases of mitosis, which basically is broken down into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Now, remember, this is a big continuum. So that is to say, one stage flows seamlessly into the next. We just draw these for us to be able to understand them better. So let's take a look at the events of prophase. In prophase, there's a couple of major things that happen. At this particular point, S phase has already happened. The chromosomes have already been duplicated. So a couple of big events that happen in prophase. Number one, your nuclear envelope breaks down. That is the envelope that covers the DNA. The nucleolus disappears. The chromosomes become condensed and are now visible. They kind of look like gummy worms at this stage. Thick and short. And your spindle fibers appear. You actually get migration of the spindles to the antipolar position. So these are all events that are happening in prophase. So here you can see in early prophase, you've got your spindle poles, right, into the centrosome that were duplicated in G2. They're now going to be migrating into an antipolar position. And so now you can see how your spindles are getting further apart, and then eventually they chain their antipolar position like this with a fully established spindle. Now, early on, you're going to notice that you basically dissolve your nuclear envelope is disappearing. And then what's going to happen is the spindle is going to attach to the centromeres of each of the chromosomes on either side, so that you have one pole attached to one side and the other pole attached to the other. And so this is kind of what they look like. So here's an early prophase where you can see the chromosomes are condensed. You can see they're kind of fat and pudgy looking, like little night crawlers or little gummy worms. Here, the nuclear envelope has basically been broken down. So it would have been right around here, but it's gone now. You can also see in this particular case, the 2 spindle poles have acquired their antipolar position. And then you can start to see that these chromosomes are getting attached and they're starting to get lined up along that midline. And those are all events that are happening in prophase. Now, prometaphase basically is where the spindle fibers are attaching to the centromeres, and they tend to shorten and thicken, and then ultimately they align along the midline. And this is called the metaphase plate. So that's all happening in prometaphase. And then ultimately, the final destination or the final outcome of prometaphase is metaphase. In metaphase, basically, you have what's called a metaphase plate, which is this sort of fictitious line of perpendicularity that draws the axis between the 2 poles. And the chromosomes are aligned along this metaphase plate. This is kind of what it looks like. So here you can see a metaphase cell where your pole, your metaphase plate, is basically this little line of central tendency that sort of lines up and aligns the chromosomes between the 2 poles. And so here you can see on this image, the line of central tendency is basically lined up between your 2 poles, and you can see your spindle is well developed here. And so all of this here, these fibers here, these are all the spindle, the parts of the spindle. Then in anaphase, what happens is essentially your centromeres divide. So basically, the sisters are separated to both poles as the poles basically pull them toward themselves. And so here you can see the 2 poles then start to pull the chromosomes toward each other, right? So you have half of the sisters going to one pole, the other half of the sisters going to the other pole. So you can see how these guys are going to this pole, and these guys are going to this pole. That's anaphase. And then in telophase, basically you have the finishing of that process. So at this particular point, you start to see the sort of reverse of prophase. So for instance, the spindle starts to disappear, the nuclear envelope gets reconstituted, the nucleoli start to come back. The chromosomes, they start to become more indistinct. That is to say, they're decondensing again. So you're unraveling them. So you're loosening them up, unpacking them, so to speak. And that's what this final stage looks like. So you can see you're reconstituting the nuclear envelope. You still have a centrosome, but the spindle is now gone. And what you end up seeing here is basically two tight nuclei in either side of the cell that are now ready for cytokinesis. And cytokinesis basically is the division of the cytoplasm of the organelles. In animals, it tends to occur by what's called the cleavage furrow. Essentially, what you do is you pinch apart the cell. So in the middle, there are actin filaments that form a contractile ring. So basically that contractile ring will kind of be this like little ring that's like a cinch cord. You just basically pull on it and the ring will become smaller, 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 and then a cleavage furrow starts to pinch that cell in half. So here you can see the cleavage furrow in this area right here where you can see this contractile ring starts to get smaller and smaller, smaller. It's attached to the plasma membrane. So as it gets smaller, 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 it starts to pull that plasma membrane in, and eventually it's just going to pinch it apart. And then you'll have two new cells. Okay. Now, let's take a look at a related process, but a little bit different. So in meiosis, as opposed to a duplication type of a division, this is a reduction division. So ultimately, in meiosis, you have two consecutive cell divisions without an interphase in between. And so in this case, your goal is to produce 4 daughter cells, not 2, and each of which is a haploid. So for instance, the parent cell is a diploid in meiosis. You go through 2 phases, broken up into meiosis 1, and then back to back, second round, which is meiosis 2, and your ultimate outcome is going to be 4 haploid cells, which now have half the number. We refer to it as haploid. So where diploid had twice the number of chromosomes, a haploid only has 1. So here you have 2 sets of chromosomes. In the haploid state, you only have 1 set of chromosomes. Designated as 2N and N. So ultimately, that's what the outcomes are of meiosis. So different. So notice now what you have are genetically non-identical daughter cells that are haploid. And we'll see why that has to be here in just a second. But ultimately, the important thing about meiosis is its key is that it basically introduces into the human genome genetic variation. So because each cell is genetically different from the parent, then that means you have new combinations of genetic material that can be different from the parent. So typically at the start of meiosis, what happens is your diploid parent cell and the chromosomes that are in them occur in pairs. That is, one from mom and one from dad. Each pair is referred to as a homologous chromosome. That is to say, one from mom and one from dad, right? So ultimately, when you take a look at it, like if you were to take a look at chromosome 1, the homologs for chromosome 1 would be chromosome 1 from mom, right? This would be mom 1, and then chromosome 1 from dad. This would be dad 1. The two of them together are homologs. They look alike, they carry the same genes, but they have different sequences. So that is to say that even though on chromosome 1, we may have the gene for eye color, mom may have blue eyes, different sequence, than dad who has brown eyes, different sequence. And so this is kind of what it looks like. So typically speaking, what happens is in meiosis 1, you're going to have a diploid cell, which is going to go through meiosis 1. It's going to separate the homologs from each other. So the duplicated homologous pairs will then basically separate into different piles. So what happens from here is you go from diploid to haploid because you're separating your DNA content. Then in meiosis II, looks very similar to mitosis, in which case you're going from haploid to haploid. And so this basically is identical, meiosis II to mitosis. So the real magic in meiosis is happening in meiosis I. So in meiosis I, DNA's been replicated, and then during meiosis I, actually during prophase I, what's going to happen is your homologs are going to find each other. They're going to pair. So that is mom one finds dad one, right? So this basically, they come together, they line up side by side, and then they synapse together. That is to say, they're zippered together with a structure that basically will kind of zip them, called the synaptonemal complex. It'll basically take mom one and dad one and kind of zipper them together. Keeps all four of those chromosomes close during the first phases. But in this particular case, there is no extra replication. You just go from meiosis I into meiosis II, right? So the idea here is basically now in meiosis I is you separate your homologs into two different piles. This is kind of what it looks like in a little bit more kind of a prettier version. So here you go through DNA replication. So now you've got the replication of each of your chromosomes. You've got your homologs, which find each other and they zipper together. Then in metaphase I, they align in double file, and that's an important piece. So what's going to happen now is the metaphase plate, which is this little dotted line that I'm putting in there, this becomes a line of demarcation, the line of separation. So once anaphase I hits, then basically it pulls each of the homologs toward the pole, and then telophase I will basically reform the cells around it. So you'll have now basically two cells that are haploid in nature because you've separated your homologs from each other. Then in meiosis II, it's exactly the same as mitosis. So let's go ahead and take a look at it. So in meiosis II, you're basically going to have everything times two, right? Because now you have two cells instead of one. So in meiosis II, you're going to line up again in metaphase. You're going to line up in single file, just like in mitosis. Those sisters then are going to get pulled apart in anaphase to each pole. And then you're going to have four different, genetically different cells. And those are your daughter cells. Okay, so basically you have kind of all the differences happening in meiosis I, and you essentially have a mitosis-like division in meiosis II. So where does the genetic variation then come from in meiosis? Well, the genetic variation essentially is caused by having different combinations of genetic characteristics from each parent. And so ultimately, they have the same phases, but where does the difference come from? Well, let's take a look at it. Meiosis I, you got prophase I. Now, the key things happen in prophase I are all the same in mitosis, with the exception of a couple of extras. For instance, synapsis is something that occurs, and homologous pairing, right? This is something that unique. These two are unique to meiosis I. Also unique to meiosis I is what's called crossing over. This is when two non-sister chromatids exchange genetic information. And so it kind of creates a mixing up of DNA material. And so this is kind of what it looks like. So here you can basically see the sisters that are kind of like interchanging with each other. Notice the mom ones and the dad ones are basically interchanging with each other. So what ends up happening is these guys will swap. So here's the regions of swap. These little crossover events, these are what's called chiasma. They're chi because they're like X-like structures. And these are areas of exchange. So for instance, if I were to follow this chromosome, I would go down here. As soon as I hit a chiasma, I have to cross over to the blue one. And when I hit another chiasma, I have to cross over to the red one. So this one starts off as red, then goes blue, and then ends off as red again. The other one starts off as blue, crosses over to the red, and then ends up as blue again. So it's just the opposite. So you can see now how you basically have 4 genetically different types of chromatids. And this creates a lot of shuffling. Shuffling of DNA and genes. That creates a lot of genetic variation. So in metaphase 1, the homologs, which are paired, they align at the equator. And as they align at the equator, basically it's a 50-50 chance as to whether or not either mom or the dad chromosome is aligned at any given point. So for instance, how do I know that my homologs will align as mom on the left and dad on the right? Well, this is a 50-50 chance because I could equally get dad on the left and mom on the right. This is also a 50-50 chance. So which one goes to which pole is a little bit of a coin flip. That's what's called independent assortment or independent alignment along metaphase 1. So for instance, I could get any one of these patterns in addition to that crossing over that's already happened in prophase 1. But see here, I could basically line up as mom, 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 or I could do mom, mom, dad, or I could do mom, dad, mom, or I could do dad, mom, dad. So any of one of these patterns can basically occur as well. That creates extra genetic variation. And so meiosis is what underpins the production of gametes in humans. For instance, spermatogenesis will make sperm in males, oogenesis will make eggs in females. And so ultimately, in meiosis, these daughter cells will then become the gametes, the fully functional gametes. So in spermatogenesis, typically this is something in males that are happening pretty constantly. So about 300,000 or so sperm are made per minute, 400 million per day. So there's a lot of sperm. It's a lot of it is just use it or lose it. You're constantly making sperm. The primary spermatocytes typically are the diploids. So what happens is these guys are the ones that will go through meiosis 1, and they'll create the secondaries. So the primaries, which are diploid, will create the secondaries, which are haploid, right? Because their homologs have been separated. And then after that, the secondaries will divide during meiosis 2 to create the 4 spermatids. So the spermatids are the 4 genetically non-identical haploid cells of the sperm. And then basically each of these spermatids will mature into sperm, spermatozoa, and then all 4 of the daughter cells will become sperm and they'll be used. So for instance, here's your primary spermatocyte, which is going to go through meiosis 1 to create your secondary spermatocytes, which are haploid because you've essentially separated your homologs. And then in meiosis 2, you're separating your sisters to create your 4 genetically not identical spermatids, which will then develop and grow into sperm, each of which can be used for the purpose of fertilization. In oogenesis, you have the same basic meiotic situation. In this case, you have a primary oocyte, which is the diploid in nature, and it divides during meiosis 1 into 2 haploid cells, either the secondary oocytes. And so this then becomes arrested in metaphase 2, and it doesn't complete that metaphase 2 unless the sperm fertilizes it. And so the only one of these cells is chosen to be the egg. The other one becomes what's called a polar body. And so oftentimes it'll just be kind of, we'll just kind of ignore it and it'll just fade away. And so during oogenesis is kind of what it looks like. So essentially the primary oocyte will go through its first division and it'll rest in meiosis 2. And so you'll have sort of the first polar body that's just kind of hanging out there. And then the sperm will come in and fertilize it. That will basically trigger the egg to complete meiosis 2 and fuse with the sperm nucleus to create the newly established zygotic diploid. The other cell is another polar body. So those will be retained. In some cases, those become endosperm and different types of nutritional things for the early embryo in some animals. So the oogenesis in females is a little bit more complicated than spermatogenesis in males. And so what happens is the secondary oocyte of the egg basically will leave the ovary. It'll be extruded into the fallopian tube or the uterine tube. That's the ovulation process where it'll hang out and wait until it's fertilized. And so ultimately, once it's fertilized, then it'll be activated to complete that second mitosis. It'll fuse with the male pronucleus, and then eventually it'll start going through embryonic division as it tumbles its way toward the uterus. And we talked a little bit about that in the last chapter. So the important thing about meiosis is essentially is you keep the number of chromosomes constant from generation to generation. Instead of doubling and dividing, what you're doing is you're halving and adding. And so that allows you to be able to add your chromosomes to another individual without increasing the number of chromosomes every generation. It also will provide a mechanism for genetic recombination, which generates genetic variation. And that's a good thing. And that's coming from crossing over and that independent of chromosomes around the metaphase plate during metaphase 1. And this will create a lot of diversity for us to be able to survive lots of different challenging conditions of the environment that change constantly. So that's kind of what mitosis and meiosis look like side by side. So here you can see in prophase 1, you're going to have condensation and spindle pole migration and nuclear envelope breakdown, just like you do in prophase of mitosis. The only difference is here now you get homologous pairing, synapsis, and recombination, where you don't have any of that in mitosis. In metaphase 1, you get alignment of your homologs at the metaphase plate, whereas in metaphase of mitosis, you get alignment of your chromosomes in single file along the metaphase plate. In anaphase 1, you separate your homologs to each side, basically making you a haploid. In anaphase of mitosis, you're separating your sisters to each side, maintaining your diploid integrity. In telophase 1, you reconstitute your nuclear envelope. You basically decondense your chromosomes, and you wait until meiosis 2. So you're only halfway there. In telophase of mitosis, you're pretty much done. So all you have to do is basically just reconstitute the nuclear envelope, and then get ready for cytokinesis, and you're going to be 2 separate cells. So in meiosis, replication only takes place once. So you don't do a replication in between meiosis. And also, meiosis is 2 stages, right? 2 divisions. Meiosis will produce 4 non-identical daughters. Meiosis will produce 2 genetically identical daughters. And also the daughters in meiosis are haploid, whereas in mitosis, they're diploid. The daughters of meiosis are non-identical. We just mentioned that. And then the nuclear divisions are different in terms of occurrence and process because you have 2 different pieces. So occurrence, basically, this is when your reproductive organs are occurring. So they're only occurring in your reproductive organ and only for the purpose of gametes. So it's not something that's happening everywhere. Meiosis is in its occurrence, whereas mitosis is more common. It's basically everywhere throughout your body. It's through tissue growth and also repair. So it's much, much more global. Now, the homologous chromosomes will pair. That's one of the prophase 1 differences between meiosis and mitosis. And so they have alignment, that's synapsis. So this is basically talking about synapsis. That's also an event that happens that's unique to meiosis in prophase. Ultimately, they will move the homologs to each side as opposed to the sisters, which is in mitosis. And these will be what you're separating. So you're kind of separating slightly different things. So for instance, the difference between meiosis 1 and mitosis is incredibly extreme. So this is kind of a side-by-side for you table folks who like to sort of see your differences between the 2 side by side. So the events of meiosis 2 are pretty much the same as mitosis, pretty much spot on. So you can see how they're the same. So you notice that they're pretty much identical for the most part. You're separating the sisters in both situations. Now, let's take a look at inheritance of these. So for ultimately, an individual with 22 pairs of autosomes has 44 chromosomes, and they have the 2 sex chromosomes, either XX or XY, depending on whether they're male or female. And so each gene will have the traits that we necessarily need for our particular, for the way we look, right? So for the color of our hair, the color of our eyes, things of that nature. But these genes can be a little bit different from each other. We call these differences between the genes alleles. Like, for instance, if I have a gene, which is maybe controlling eye color, I can have different versions of that gene, like different colors of eyes. Like, for instance, I can have blue versus brown. Those will be the alleles. And so ultimately, changing the number of chromosomes and also the combination of alleles for a trait can kind of change the way things are expressed. And generally speaking, these have very negative effects in most cases. For instance, in animals, and us specifically, we do not tolerate extra DNA or lost DNA very well. So we can't have too many or too few autosomes. That's just something that we don't tolerate well. When this does happen, it's usually caused by a process called non-disjunction. And so what this is essentially an error in segregation. And this causes usually what's called asymmetrical segregation, lopsidedness. Okay. Now, generally speaking, during meiosis 1, if you have a missegregation there or lopsidedness, usually that means that the homologs are failing to separate correctly. If it's a meiosis 2 non-disjunction, that means the sister chromatids are failing to separate correctly. What this can lead to is basically a situation where you have too many chromosomes or too little. If you have too many, we refer to it as a polysomy. In this case, a trisomy is specific, in which case you have one type of chromosome that has 3 copies. So we refer to this as 2n plus 1. So 2n is our normal, that's our 2, plus an extra 1, which is the 1. If you have too few, we refer to this as monosomy, where you only have 2n minus 1. So basically 2n is our normal, we take 1 away, that means we only have 1 chromosome. So this is basically 3 chromosomes, that's why we call it trisomy. And this is 1 chromosome. That's why we call it monosomy. So ultimately, if we have 22 chromosomes, then in a 2n plus 1, that means we will have 23 chromosomes. That's without considering the sex chromosomes. And it's kind of what it looks like. So here's a situation where we have a meiosis 1 non-disjunction, right? So this is a non-disjunction event during meiosis 1. Normally what's happening is these 2 homologs are going to separate. And when they separate, they're going to go to each side, right? That's what's supposed to happen. But what happens in non-disjunction is all of them go to one side and nothing goes to the other, right? Now, what happens after this is typically meiosis 2 goes well. And so what happens is you're going to get 2 chromosomes in each of these, and you're going to get nothing in each of these. Now, when they get fertilized by a sperm, the sperm is going to add its chromosome, giving it a 3rd chromosome. Same thing to this one, giving it a 3rd chromosome. So these guys are trisomies. Now, if the sperm fertilizes these eggs, it's going to add its 1 chromosome. So you only have 1 chromosome. These are monosomies. So in an M1 non-disjunction, you typically are going to have either too many or too few. Now, what happens in an M2 non-disjunction? When an M2 non-disjunction, typically the first division goes normally. For instance, your homologs are separated normally to either side. So you can see one over here and one over here. Now, what happens is usually one of the 2 non-disjoins. It's rare that both of them do. So, for instance, let's take a look at the normal pattern. So the normal one, the normal meiosis 2 is going to occur normally. So each sister is going to go to each side, and you're going to get 2 healthy cells with your 1 chromosome. When it gets fertilized by the sperm, it has 2 chromosomes like it's supposed to have, and the other one is also going to have 2 chromosomes like it's supposed to have. This side is going to be normal offspring. However, in the non-disjoined side, this is an M2 non-disjunction. Everything goes to one side. So everything basically got pulled over the other side. The other one has nothing in it. So when it gets fertilized by the sperm, then you're adding a 3rd chromosome, which basically creates a trisomy. On the other one, since you don't have any chromosomes there because they all went to the one side during non-disjunction, then the sperm is going to add its one chromosome. So this is going to be a monosomy. So in the case of a meiosis II non-disjunction, in this particular case, you could expect at least half of the offspring to be normal. Whereas in a meiosis I offspring, both of them are going to be abnormal, trisomy or monosomy. Probably, more than likely, this individual will probably experience a high rate of miscarriage, because a lot of times first trimester miscarriages are mostly because of abnormalities in chromosome number. So an abnormal number of autosomes typically will create a lot of abnormalities. Monosomies of pretty much all the chromosomes are pretty lethal. They'll never make it to term. Trisomies also are very lethal. The only one that has any kind of penetration is trisomy 21 Down syndrome. And so that has the biggest chance of making it to birth. They can actually live a fairly decent life, but their life expectancy is oftentimes limited. Why? Because their life expectancy is like maybe in their 50s, as opposed to like in their 80s or 90s, depending on who you look at. So taking a look at further changes in chromosome number, one of the things we notice is if you happen to have a trisomy or monosomy in the sex chromosomes, it's much more tolerated. A female is XX and a male is. And so what a female typically does is usually to kind of match up with a male, right? Because the idea is, I mean, a female would have twice as many X genes as a male. You would expect that to be a bad thing. It's a duplication, right? So what a female does is actually she takes one of these X's, and it's a random selection, and she inactivates it and kind of sticks it off to the side of her cell. And we can see this cytologically as what's called a bar body. And so typically a female cell will function with a single X chromosome, just like a male does. And that's the reason why she can escape this problem of having too many chromosomes, because she inactivates one of them. And so this is the reason why, say, for instance, somebody who has an XO, that's Turner syndrome, is able to survive, because they only need one X in order to be able to do what they need to do. So ultimately, regardless of how many you have, the extra X chromosomes will basically be condensed down into bar bodies. So for instance, if you have a female, or excuse me, if you have a male who's like XXY, this is survivable ultimately, because these are essentially a situation where you can inactivate some of these and sort of stick them off to the side. If you have an extra Y, it's not a problem, because there's really not that many genes on the Y. So you can actually inactivate one of these. If you have a triple X female, for instance, you can inactivate these. And then that gives you your 1. And you can inactivate one of these X's to basically make you a functional XY. Now, generally speaking, whenever you get an XYY, that's going to be a non-disjunctional event in spermatogenesis. A lot of these other non-disjunctional patterns are non-disjunction happening in females. So the most viable of all of our trisomies is Down syndrome, right? So this is basically a situation where you have an extra 21, and this increases in frequency the likelihood for mothers who are over 40. As a matter of fact, if you're over the age of 35, a lot of times you will be automatically categorized as an at-risk pregnancy because of this increase in likelihood of trisomy in the mother. And so oftentimes what will happen is with the Down syndrome, you'll have short stature, eyelid folds, flattened facial profile, things of that nature. And so a lot of times they'll have developmental delays, intellectual disability, things of that nature, to varying severities. And they'll typically have a shortened lifespan as well than would be normally the case. Now, this is because of an extra 21. That's usually an aneuploidy associated with, an aneuploidy is a general term for all increases or all extra or missing chromosomes. It's a general term. Typically, it's in the mother. You see this one as a non-disjunctional event of the mother. And so generally speaking, it's oogenesis. Most non-disjunctional situations are usually in the mother. There are some that are spermatogenesis. These are less common. Why? Because with males, they're constantly making sperm. It's use it or lose it. If they have a bad non-disjunctional type of a batch of sperm, chances are they're just going to scrap that and they're going to just make a new batch, and that new batch will probably be okay. Whereas the female, the egg that she selects is something that she's been hanging on to potentially for 4 decades. And the idea is that the reason why there's this maternal age effect is because they think that whatever the mechanism is that kind of keeps those eggs in stasis begins to break down as the mother gets older, and it creates more opportunities for non-disjunctional errors, which is the reason why we see a lot of non-disjunctional patterns coming predominantly from the mother as opposed to from the father, although they do occur in both, just not at the same frequency. So this kind of creates a general type of a syndrome sort of a thing. And a syndrome, definitionally, by a medical definition, is just simply a group of symptoms that always occur. It usually means we don't have any clue whatsoever what causes it or how to define it. It's typically just a general kind of observation of a group of symptoms that seem to be occurring more than just randomly. So the presence of the Y is what determines male. The reason is because on the Y is a special gene called the SRY, the sex determining region of the Y. That's what this actually stands for. And what this does is it actually holds a gene that basically makes what's called testis determining factor, which is an important molecule that actually induces masculinization of tissue. It causes the male sex organs to develop. In the absence of testis determining factor, the female will develop. So the Turner syndrome, which is XO, only has one X chromosome, typically is short, has a webbed neck. The reproductive organs, uterine tubes, ovaries, uterus, are typically pretty underdeveloped. Their breasts oftentimes will not develop. And so they can have intellectual disabilities, but they can be like trisomy 21, Down syndrome. They can be live fairly normal lives. Klinefelter male, which is typically the alternative to Turner syndrome. This is an XXY male. So it has 2 chromosomes. Generally speaking, the testes and prostate gland are typically underdeveloped. So there's a little bit of feminization of the tissues. For instance, breast development may occur, things of that nature. So they tend to be slow in learning, which is difficult to capture and quantify. But they don't have the same kind of intellectual disability that others would have. They do have an increased risk of breast cancer and osteoporosis and lupus, which are things that are true for women. So you can see that having the extra X kind of feminizes what would otherwise be a male body. And so the medical treatment is to treat with testosterone. So that's the masculinizing hormone that essentially masculinizes male tissue, the male body. It also increases muscle mass and things of that nature. So it kind of counteracts the feminizing effects of the extra X. Now, a poly X female, like triple X, quadruple X, basically all these extras will just basically be inactivated. So the extras will basically be inactivated. And so ultimately the female should be, for the most part, mostly normal in terms of appearance and things like that. There may be some subtle, very small, delays and things like that, but nothing perceptible to see like, yikes, there's a situation going on here. Jacob syndrome, which is an extra Y. This is typically a non-disjunction of spermatogenesis. So this is the dad doing this one. He passed on an extra Y when he's only supposed to pass on one. So they usually tend to have a few problems. So they tend to have, for instance, a little bit of speech and verbal delays and things of that nature. And so, but they should be fertile just like anybody else. So they just have to give a Y or an X, and they can certainly do that. So ultimately, then, when you take a look at changes in chromosomal structure, you're talking about things like radiation or mutagens, basically things that can change or alter your DNA. And so normally these are things that will cause issues. So like breakages in DNA that become an issue, broken ends and things like that can all create problems. And so some of these changes can include things like deletions of DNA material, duplications of extra DNA, inversions, and translocations. And so the deletion piece is where a piece of the chromosome actually breaks off. And so it's essentially is a loss of information. So you lose some information. If you lose genes in there, then they're gone. And that could be a very bad thing. And so here's an example of a deletion. So we have a chromosome A, B, C, D, E, F, G. In this case, we're going to delete A. So A should have been right here, but it's gone now. So it's deleted. So that's a deletion. In a duplication, this is essentially where you have a chromosomal segment that occurs more than once, sort of like a stutter, right? So where you kind of stutter in the chromosome and you add an extra piece. So in this particular case, when you take a look at A, B, C, D, E, you're going to basically duplicate DE, and then that's going to be kind of like a little bit of a stutter. So where you have a back-to-back duplication, it adds extra material. Now remember, these basically act like micro losses and additions of DNA, right? So basically, this is an addition of DNA. Now, this is the same problem, by the way, we're running into that we had with chromosomes, where we said you can't have an extra chromosome or missing chromosomes and it go well because we don't tolerate additions or subtractions. And this is micro additions and micro subtractions. So these can also cause varying degrees of problems. In an inversion basically is a situation where the chromosome literally is flipped around 180 degrees. Oftentimes they don't present any problems, but they can, right? So in an inversion, you can see you have A, B, C, D, E, F, G, and then you have A, B, and then instead of C, it's CD, you flip it around and you say DC. So you basically just inverted it. You pop this out and you spun it around 180 degrees and put it back in there. So that's an inversion, but it's still mostly there. And then a translocation is basically where two genes swap genetic information, right? So this is where two chromosomes swap regions. And so you have different areas, like for instance, in 5% of cases of Down syndrome, this is an area where 21 and 14 have kind of swapped in a translocation event. And so here you can see you have two different chromosomes, A, B, C, D, E, F, G, H, and then L, M, N, O, P, Q, R. And then what's going to happen is I'm going to swap these two regions right in here. I'm going to break one off of the other, and I'm going to basically put it and swap them back and forth. So now I see the QR section on the A, B, C, D, E, F gene, and I see the GH region on the LMNOPQ or the LMNOP gene. So you can see I basically swapped these guys back and forth. That's a translocation. And so this is kind of what it looks like. So essentially what happens is when you have an inversion, it creates problems on pairing, right? So basically your homologs are a problem because they pair by their identity. So in this particular case, what happens is you want to pair your A's with your B's and your C's with your D's and your E's with your F's and so forth, but it can't because it's inverted. So when you have the inversion, then the E wants to come over here with this E, and the C wants to come over here with this C. So what it does, it creates this little inversion loop where one chromosome kind of flips around so that it can get C to C, D to D, and E to E. And by doing that, it's basically able to sort of pair, but it's not really all that great. So if you have a regional crossover in here, then you can start to cross over all these different crazy things. So now you have A, B, C, D, E, B, A. So it's kind of like a weird combination. And you've got G, F, C, D, E, F, G. So you can see you're starting to get some crazy combinations because you're trying to manipulate your way around this inversion because it's not really pairing up very well. Now, what are some deletion syndromes? Well, Williams syndrome, right? This is basically a chromosome 7 loss. So a couple of things you'll see, and these are all very generic. What happens here is the gene for elastin is missing. So you turned up nose, wide mouth, small chin, large ears. You'll have learning disabilities oftentimes here, but you have really, really good musical abilities and verbal abilities. So you kind of have like an accentuation of one and a depression of the other. So the gene for elastin missing will affect your cardiovascular system, your skin, and allow you to age prematurely. So the elasticity of your blood vessels probably won't be so great, and that'll be probably cause a lot of cardiovascular problems. Um, Friendly, outgoing temperament, like I don't know what that means. I mean, so if you're not a Williams person, you're just an angry person. So you can see some of these are very, I mean, you kind of have to sort of go with it a little bit. They're not, super quantitative. But these are just some things that we see. Creta-Shaw basically is where a piece of chromosome 5 is missing. The important thing here is that you will have intellectual disabilities with this one, some facial abnormalities. Also, your vocalization, part of the problem is there's abnormal development around the larynx. So your vocalization will sound like a cat, and that's where they get the Creta-Shaw, which actually is French for the cry of the cat. Some translocation syndromes, basically where you essentially are inheriting these translocated proteins. Alagille syndrome is basically a translocation between chromosomes 2 and 20. So you have different heart defects in this one. You have some clubbing, widening of your fingertips may also occur. They can also occur, translocations can cause cancer, depending on where they land. So it all depends on the nature of the break. Each translocation is different. So we can't say they all do. We can just say we have to study each individual translocation to figure out what specifically it actually does. And so that is basically chapter 19.